Okay, thanks. Can you hear me?
Yes, very well.
Good. Except my slide isn't moving. Huh. My screen is frozen. Yeah, my screen is completely frozen. I can't move it. Let me.
Do you want to try pressing escape, getting out of the?
I've tried. It's not moving. Let me just. There it goes. Okay.
Okay.
Okay, so there are a lot of questions that many of you are already aware of that we don't know, things that we don't understand about COVID pathology and the optimal therapies. So we really don't know anything about the viral load in determining the pathology, the role of the initial innate immune response, the role of the adaptive immune response in controlling the disease. And then there's good evidence that an overexuberant innate and adaptive immune response may be driving the ultimate lung pathology. Furthermore, in thinking about all of this conundrum actually means that the timing of various immune boosting and immunomodulatory approaches is going to be very, very critical because we have a bit of a yin and a yang going on here. And so to work out these issues of timing, animal models are what we need. And there are several animal models of COVID-19 disease, the ferret, the Syrian hamster, but those immune systems are clearly quite far from the human. The monkeys are much closer, and those are great models, but they're not all that nimble because you can't really test large cohorts and look at the timing of different therapies in a nimble way. And then there are the human ACE2 transgenic mice with murine immune systems, which are extremely useful, but they're still immune systems far from the human. So what we want to do is use human immune system mice that will allow us to study the human immune response and to adapt these models to allow them to study, be used for a study of SARS-CoV-2 infection. So human immune system mice, that's a broad category of models. There are many variations. Pretty much all of them involve immunodeficient mice. The ones that are used most commonly because they're most efficient for allowing human reconstitution are the NSG, which are deficient in T and B cell rearrangement, can also be a RAG knockout, and in the common gamma chain that is needed for IL-2, IL-7, IL-15 signaling, and these mice are completely deficient in TB and NK cells, and then therefore quite permissive for human cell engraftment. Now, one way that this is done is to inject human PBMCs into lightly irradiated NSG mice, but that will very quickly cause you xenogenetic GVHD, and that doesn't give you much of a window to do any kind of a study, although it has some utility. So the alternative is to inject human hematopoietic stem cells and let those differentiate de novo in the mouse. And then you get human B cells and myeloid APCs just by injecting those stem cells. And you'll get T cells developing in the native thymic rudiment in the NSG mice. But this rudiment is small and never becomes quite normal. I'll come back to that. So the alternative then is to, instead of letting the T cells develop in the mouse thymus, let them develop in the human thymus. And that's best done with fetal human thymus, which is a little bit limited currently by federal restrictions. So we want to adapt these models for COVID-19 research. And one way to do that is to introduce the human ACE2 into immunodeficient mice. And a second way is to provide human lung tissue into these HIS mice. And I'll come back to these models. And finally, we want to improve the lymphoid structure in these HIS mouse models to allow better vaccination responses. So this slide illustrates the immune reconstitution you get with various kinds of thymi. And Mohsen Khosravi in my lab has developed an efficient, rapid technique for thymectomy of these mice. So we can actually study mice with no thymus and compare them to mice with an intact thymic rudiment and to those with a human thymus graft. And what you see is the B cell, monocyte, and T cell reconstitution in these animals, and there are absolutely no T cells in the mice that are thymectomized. There are small numbers of human T cells in the peripheral blood of mice with a mouse thymus intact, but in thymectomized mice with a human thymus graft, that is where we get the best T cell reconstitution. And that improved T cell reconstitution also seems to enhance the monocyte and B cell reconstitution, probably by providing signals. And just to show you why the T cell reconstitution is so much better with a human fetal thymus, this is the fetal thymus graft at the time of takedown. It was 1 cubic millimeter when implanted under the kidney capsule, but it's grown quite big, and you can see the cell counts get up into the 10 to the 7th and 10 to 8th range. In contrast, a mouse with a native thymus that repopulates with human thymocytes has far fewer cells in it and is much smaller. Okay, so the models that we're studying are several. So one involves thymectomized MSG mice, and then they can get a human thymus graft, fetal thymus, and we're trying to also improve the use of neonatal thymus to avoid fetal tissue for federally funded research. So this thymus is put under the kidney capsule. We inject human hematopoietic stem cells, and we can do that from several sources. We've done it with all three, and again, we can avoid fetal tissue using cord blood or adult bone marrow. So the modification that we're now working on to put some lung in, there are two. One is to put fetal lung tissue from the same donor as the fetal thymus and HSCs, and that experiment is now ongoing. This is implanted under the subcutaneously, and I'll show you what that looks like. And this is a collaboration with Wei Wang and our humanized mouse core and Yongguang Yang. And then the second will be to generate lung bud grafts from autologous stem cells, stem cell derived iPSC. And you saw last week from Hans Snook what those grafts are like, and those are put under the kidney capsule, and we're collaborating with Hans on that. And then finally, when these mice are fully reconstituted, we'll study viral pathology and the effects of the immune system, infecting the animals with SARS-CoV-2 and testing various therapeutics and vaccination approaches. Just to show you, this is some work already done by Wei Wang and Yongguang Yang, where they implanted fetal lung along with the human immune system. And you can see that these lungs actually did have CD4 and CD8 Tcells in them. And this is what it looks like subcutaneously after 22 weeks. And you can see lots of lymphoid cells in those grafts. The second model, and this HLA2 donors that can interact with the HLA2 positive lung tissue. So this is my last slide, and I think I've already really touched on most of this. The other modification we're doing is using an AAV vector carrying murine TSLP, which has been reported to enhance the mouse lymph node and thymic structures that may allow us a better model for vaccination. So thanks. Happy to take any questions.
Thank you, Megan. And at this point, if there are any questions, please raise your hands. Jahar? Jahar?
Sorry, can you hear me now?
Yes,
okay.
We can hear you.
Yeah, I'm sorry. Hi, Megan. Just wanted to know, my understanding is that when you implant a lung in a kidney capsule, a human lung in a mouse kidney capsule, the vasculature of the lung is still the mouse vasculature. And I was wondering what is known about the interactions of mouse endothelium with human monocytes that are coming from the bone marrow. Have you thought of, is there anything known about that?
It's a great question. We actually haven't done it with lung, but we have looked at islet allografts and xenografts under the kidney capsule in these reconstituted humanized mice. And what we see in those grafts when they reject is infiltration of human T cells, B cells, and macrophages into the graft. And it's very dramatic. So I'm not too worried about that. And in fact, in other studies using the model that involves a mouse, an intact mouse thymus, we actually see very dramatic human macrophage-mediated infiltrates in the mouse tissues and pathology related to that. So the human monocytes are capable of migrating through murine vessels.
I don't see any other question, but I have a question myself, Megan. So there is an increasing relevance of the TMPRSS2 pathway, which is regulated by both AR and ER, androgen receptor and estrogen receptor. I was wondering whether in the sort of fetal lung transplant, because these are very young lungs, whether there is appropriate signaling to those pathways. That appears to be potentially one reason why younger children do not get infected very efficiently. And in counterpart, whether in the actual mouse model, whether the TMPRSS2 sort of pathway is present.
Those are both really good questions, and the short answer is no, I don't know the answer, but it'd certainly be something that we could address in these models. And I should say that, once we get these adaptations in place and show that they work, I think there'd be a lot of potential collaborations for a lot of questions that people may have. And some of those may be collaborations, some may involve the humanized mouse core that we have in the CCPI.